Hello friends, welcome to Clinical Biologist channel. So today we're going to discuss some important points regarding coronavirus. So this video is purely based on education purpose. So here we are highlighting many important points regarding the coronavirus. What are the symptoms? What are the special characteristics, pathogenesis, symptoms and diagnosis part. So watch the full video and understand the pathophysiology and the structure of coronavirus. So first we'll come to see what is important feature introduction of the coronavirus. So it's basically the coronavirus comes under Coronaviridae family, and this coronavirus usually found in avian and mammalian species. Okay, so according to the studies shows the coronavirus of human and cattle are antigenically related. The study shows this one, but there is no any evidence that human coronavirus can be transmitted by animals. This is a very important point. Can remember this one, that human coronaviruses cannot be transmitted by animals. So there is no evidence till now. Okay, so this is very important point. Okay, so and also in the coronavirus in humans, it causes a mild upper respiratory tract infection. Okay, so usually here you will find like a common cold, like a symptoms and dry cough. So these are the very important points you can remember in the coronavirus. So also there is no evidence, even the study shows there is a gastrointestinal tract infection also caused by the coronavirus species and associated outbreak in children, diarrhea, but there is no any particular study shows properly there is a gastrointestinal viruses. So the basically remember, it's a coronavirus is found in avian and mammalian species. Okay, so coming to the structure part of coronavirus. So if you see the structure of coronavirus, corona, so the crown, it's a crown like a structure. So it's a spherical and pleomorphic in shape, and it's enveloped particles. Okay, so spherical in shape or a pleomorphic, which contains enveloped particles, and it is a RNA positive sense single stranded RNA virus. Remember this point, single stranded positive sense RNA, and there is a nucleoprotein which is a present in the capsid and compromised of matrix protein. This is a very important point, nucleoprotein. And also the envelope bears a club shaped glycoprotein projection. So the club shaped glycoprotein projection, what you can see the crown like a structure. Okay, that is a contains a glycoprotein projection. So these glycoproteins are very important, and it's responsible for the attachment to the host cell. Okay, these glycoprotein, enveloped glycoproteins, are responsible for attachment to the host cell when it's come in contact with the host. Okay, when it's transmitted through a close contact or fomites or airborne. So it's very important. The important point is the glycoproteins, which is what attached to the host cell. And one important here, there is a one serotype of coronavirus, so that is OC43, which possesses a hemagglutinin on their surface. Hemagglutinin. So this is one important. So there is a two important serotypes. One is OC43 and other one is a 229E like serotypes. So here OC43 is possess hemagglutinin. Coming to classification, so this classification of coronavirus is based on mainly is a crown like or halo like appearance. Okay, crown or halo like appearance, so which contains envelope glycoproteins. We discussed before what the envelope glycoproteins will do, it attached to the host cells. And also this coronavirus comes under a Coronaviridae family. Remember this point, Coronaviridae family. So sometimes they will ask in the MCQ questions. The family belongs to coronavirus is a Coronaviridae, and these are two important serotypes of human coronavirus. That is, one is OC43 like virus, and another one is 229E like serotypes. These two serotypes is very important. Based on these serotypes, we can diagnose a particular infection of coronavirus. So 229 like a coronavirus is usually isolated in human embryonic fibroblast cultures. This is very important point. Human embryonic fibroblast cultures, because this coronavirus is difficult to isolate, and that's why it need special cells, special cultures. So human embryonic fibroblast cultures is used to isolate 229 like virus, and OC43 like virus, which can isolate or adapted by using in suckling mouse brain. So these two important characteristic, remember. And also if you see, there is a two in these two serotypes, there is a little antigenic cross reaction between these two serotypes. Okay, coming to multiplication, so how it multiply? Okay, so once we are seeing this virus is shedding from the nose or shedding from mouth or coughing by touching by close contact, once it transfer from one host to another host, the first what we'll have to do, when it attached to the host cell by the glycoproteins, remember we have discussed, and once it attached with these glycoproteins, it uncoated the genome, which is transcribed and translated. Okay, so this mRNA mRNA from a unique nested set sharing a common a 3 prime end. This is a very important. Here it always share a 3 prime end, and the new virions from the host cell is emerged by the budding. Okay, by budding from host cells, the membrane is emerged. But here the two important things you can remember, which can helps for the for the transcribed or translated, these two important points here: aminopeptidase N and salicylic acid containing receptors have been identified to act in such role. Okay, so here these two, aminopeptidase and salicylic acid, helps in this mechanism. Okay, so for 229E like coronavirus or OC43 like coronavirus. Remember these two things: aminopeptidase N and salicylic acid. Coming to pathogenesis, as you know what is meant by pathogenesis, how it causes, what will happen, what is incubation periods. So these all things will come under pathogenesis. So usually we are hearing it will transfer via airborne droplet or nasal mucosa. Yes, that is correct. So that's why we have to keep social distancing. We have to quarantine ourselves for once you get in such situation. Yes, if you find like symptoms like this, you have to be isolate yourself to avoid going in public places, quarantine yourself to make be safe for yourself. So it's via airborne droplet one thing, and nasal mucosa. Okay, and once it goes to the heart, it replicate and attach to the ciliated epithelium, as I told, glycoproteins, and causing a cell damage and inflammation. Okay, causing cell damage and inflammation. So what are the important two things I discussed before? One is salicylic acid and another one is aminopeptidase. These two important receptors are very important in that. Okay, so this cause cell damage and inflammation. Usually if you see, there is an incubation period is a 3 days. Okay, the symptoms will start, and if you it's look like a common cold, including a nasal obstruction. You feel like a common cold, normal common cold, sneezing, runny nose, occasionally you will find cough. So main important here, you will find the dry cough. Okay, and at the same time, that incubation period, the virus will shed in nasal secretion. So that's why hygiene is a very important. Hand wash, sometimes you have to use your scrubs, and so avoid going crowded places. So hygiene is a very important, and keep distance from such areas, and you have to isolate yourself, quarantine yourself. So that's why keep we are telling that one. So this is the one measure you can avoid and you can save with this virus. As we discuss, culture is very difficult for this. So we have told you the important culture, what or how we go into culture. So viruses are extremely fastidious. So remember this point, fastidious. Okay, and it's grow only in differentiated respiratory epithelial cells. So special cells required to grow this media, as we discussed before. And once it grow in that media, so you will find a cytopathogenic effects. What cytopathogenic effect you can see in the lab? So that is, it becomes a vacuolated, shows a vacuolated and shows damaged cilia. So that we call as a form syncytia. Okay, so that is a pathogenesis you can see when you see in the laboratory. So very important this one. So once it attached to the host cell, okay, so it triggers the production of inflammatory mediators. The virus, the mechanism inflammatory mediators, it triggers, and they which increases nasal secretion and cause local inflammation and swelling. So this is the pathogenesis which interact with the coronavirus. So remember this point, inflammatory mediators and secretion, nasal secretion will increase, cause local inflammation and swelling. So which in response, it stimulates sneezing, obstruct the airways and raise the temperature of mucosa. Okay, so this pathogenesis you will see in such coronaviruses positive cases. Okay, so coming to host defenses, what is the, how can we defend our body ourselves? So what is the host defense? So first thing, you know that our nasal secretions has a many antibodies, and even in our serum also, so which can naturally defends our body for the such infection. So mostly it's depend upon the age factor, so immunity, so that's a very important. So sometimes one third of a carrier, we call it carriers, so person can get infected with the virus and it won't show the symptoms, that we call as a carrier. So at such situation we have to take care of ourselves, and we have to be very careful for the carriers to spread the infection. So protect, so even the interferons can protect the infection. Remember this one point, interferon. So what are the interferons? We will discuss in some other topics, one important topics. So coming next, so many individuals have a specific bodies in their nasal secretions, as I discussed, many antibodies which can protect against the infection. And also there is a, in such antibodies are present which can neutralize the infectivity of the virus. Okay, so projection, surface projection, which can neutralize the infectivity of the virus. So here, so the antibodies, interferons, the antibodies present in the serum or nasal secretions in our body can prevent the infection. That is called a host defense. So defending ourselves, and that's why we have to always have to build our immunity good, and we have to protect ourselves, so avoid crowded places. Coming to epidemiology, so epidemiology, as you know, so which season it will spread, how it will spread, so that comes under epidemiology. So mainly the peaks in the winter season. Okay, so mainly the peaks in the winter season in the form of local epidemics, sometimes of few months or few weeks. And also some serotypes which can be, the study shows that the serotypes may be written after several years also. So that is a very important. So this virus is transmitted via inhalation of contaminated droplets, and also it transmitted by the hands and mucus of the nose or eyes. That's why always wash your hands and maintain hygienicity and maintain social distancing. So coming to diagnosis part, this is one of the very important part as part of a microbiology point of view. So diagnosis, so see, there is a common cold. We can see the colds caused by coronavirus. It's very difficult to distinguish between the other colds also. That's why we put on the laboratory diagnosis. We will pressure on to the laboratory diagnosis. We give the importance to laboratory diagnosis. So here, very important to detect the antibody titers in paired sera. Okay, the antibody titers in the paired sera, and I told the serotypes of coronavirus. And as I told also, the virus is difficult to isolate. So these are the one rapid things we can diagnose by the titers, antibody titers. And there is a new hybridizing nucleic acid hybridization test that is a very important and sensitive test, that is we call as a PCR also. We can diagnose by the PCR, and there is a new rapid tests are coming. Maybe it will help for to diagnose the coronavirus. Also, this is a very important, the how to collect the sample. This we can collect the nasal swab and bronchial washings for the culture purpose or for the growing isolation of virus. So this is also one of the important method to diagnosis of in microbiology. So laboratory diagnosis, as we I told, stressing that's a very important part in such outbreaks or situations. So collecting of the paired sera is very important to detect the antibodies against OC43 and 229 229E like coronaviruses, and direct hybridization tests like polymerase chain reaction, viral nucleic acid also important in diagnosis of coronavirus. So what is the control measures? How can we prevent? As always, we are making aware and creating awareness, giving tutorials, and everywhere we have conceiving. So we have to avoid crowded places, isolate yourself, make maintenance hygiene, wash your hands, because treatment of common cold is like a symptomatic. As per now, it's a symptomatic. We can give the treatment symptomatic treatment. So there is no specific vaccines till now has available. So isolate yourself, quarantine yourself, and hygiene measures reduce the rate of transmission. That's very important. Hygiene measures is very important, and create awareness for all of us. And uh, so hand wash for at least 15 to 30 seconds is very important. Use soaps. So that is one of the important to control this coronavirus infection. Hope you understand this. And this video is basically on educational purpose, and so hope this will helps you in your exams also and in your society also to maintain and to create awareness. So thank you for watching this full video, and comment us what specific topics you need to discuss, and we will try to make you new topics on emerging in microbiology or in